Hi, this is Miss Litton, and it is lunchtime, and I have some AP biologists here. Say hi. Oh my gosh, it's very depressing. And this is chapter 13, regulation of gene expression. All right, so we want to remind ourselves there is transcription, and when you do transcription, what are you making when you do transcription? RNA. Hey, look at me. Be engaged even though you're eating lunch, because it's going to be more meaningful to you if you're engaged, even if you're overwhelmed. Okay? All right. So in transcription, we're making what? RNA. Now, in order to make RNA, who are we making RNA from? DNA. So you have to have the DNA available to you, right? If you're going to make RNA. So what is it called when the DNA is tightly coiled and put away? What do you call that? Heterochromatin. So it has to be euchromatin so that we can even make any RNA. And that is one point of control. Is it heterochromatin or is it euchromatin? Okay? So now let's assume that the DNA is available. So then we want to go about transcription. Transcription is when you use the DNA to make what? RNA. How does that happen? RNA polymerase has to get on the promoter. Okay? What could block RNA polymerase from getting on a promoter? A repressor. Now, does the repressor bind to the promoter? No. But if the repressor is close to the promoter, he can kind of spread out a little bit and block RNA polymerase from getting on the promoter, right? So whether or not we have a repressor, and where does the repressor bind? What's the part where it binds onto? Operator. So when you deal with repressors, either being on or off an operator, that is all underneath the umbrella because it's dealing with a repressor. That's all about what kind of control? Negative control. Okay? Anything that has to do with a repressor getting in the way or not getting in the way, just because it's dealing with a repressor, repressed, is negative control. But sometimes RNA polymerase needs help getting on the promoter. It needs it like the DNA opened up a little bit so it makes it easier for RNA polymerase to get on the promoter. Who could help RNA polymerase bind to the promoter? A what? Transcription factor. Okay, and we looked at CAC, right, as our transcription factor. Okay, so if you need help binding on, anything that helps, I may be available for help, I may not. I may be a Boy Scout selling cookies, I may not. Or they sell popcorn. That's right, Girl Scouts sell the cookies. So, okay, so when RNA polymerase binds to that promoter, if you get help or might need help, that's not negative control, it's somebody helping you, so that's what? Positive control. That's the big difference between positive and negative control, and that was one of your stressors. Okay, and I'll show you that in just a minute. Okay, now, so now we've made the RNA. Okay, there's transcriptional control, making the RNA. Now, when we're done making the RNA, so what's a way to say I'm done and transcription has already happened? It's not, now it's called what? Transcription. Post-transcription, because I'm done making it. What are some things I do to RNA once I'm done making it? I put on a cap and a tail, and I cut out the introns before it leaves a nucleus. Anything to do with that is under the umbrella of post-transcriptional control. Why? Because we're done with transcription. Post-transcriptional control. Okay. Then what follows transcription? What step? Translation. Where does translation occur? On a ribosome. What happens during translational? And it's happening right here. It's on a ribosome. We have tRNAs landing on the mRNA, right? Anything that interferes with that, maybe a small interfering RNA, okay? Anything that interferes with us being able to do that, that is called translational control. Once I am done with translation, I will have built a what? A protein. Proteins are built out of what? Amino acids. Now, just hooking amino acids together, that's referred to in protein structure language as its what? Primary structure, right? What else has to happen to that protein? It's got to be in a alpha helix or a beta pleated sheet. That's its secondary structure. Then it gets folded due to the what groups? R groups, that's its tertiary structure. And it might have to hook up with another chain, that's its anything dealing with secondary, tertiary, quaternary structure. That is, we've already got the primary made. We did the primary structure during translation, right? Anything dealing with secondary, tertiary, quaternary, any folding of any kind, hooking up with other chains, that's following translation, so that's called post-translational control. Okay, got the big picture? Okay, so that was one of your stressors, right? Wasn't it? Translational control or something? I don't remember. Okay, so control can happen anywhere along the lines in this process. So this picture I have up right here, is that a prokaryotic cell or a eukaryotic cell? Yes. How do you know it's a eukaryotic cell? Nucleus. It has a nucleus, my friends. It has a nucleus. Okay? So this is a eukaryotic cell, and I taught you aspects of transcriptional control using a prokaryote. It's simpler in a prokaryote. So that's when we talked about this is a prokaryote, E. coli or a bacteria. And we learn the basics. We learn the basics with this one because it's so much simpler. Okay? And bacteria tend to be under the umbrella of what kind of control? Not positive, but negative. They're all about the repressors. So let's identify. Here you can see the double helix in the background. Okay? Let's identify things on here. Here's a regulator gene right here. Regulator genes, if transcribed into mRNA, and if that mRNA goes and gets on a ribosome, so it has been transcribed from a regulator gene, what kind of protein will it build? It'll build a repressor. That repressor could be built in the on position or it could be built in the off. Okay? But it's going to build a repressor. Okay, let's see what else we have. So this is farther down the chain. That's why the hash marks right here. Promoter. Who do we know likes to get on a promoter? Yeah. It's where it docks into the RNA so it can go along and read it. It's got to dock into it. So it docks in at the promoter. But what's sitting right next to that promoter? An operator. And in this case, if an active repressor is on the operator, then the full promoter is not exposed here. It's kind of blocked a little bit. And as a result, RNA polymerase cannot get on this promoter. So these structural genes right here, they can be transcribed into mRNA. Oh, weird, I'm not lined up. And then the mRNA can be translated into what? Proteins on a what? On a ribosome. Exactly. Okay, so that's weird. Okay, let's look specifically. Let's go here and let's start with the lac operon. Okay, now in the lac operon case, our regulator gene up here has been transcribed and translated. It's got an active repressor, and the repressor is on the who? Who's the repressor always get on? Operator. Okay, so RNA polymerase cannot get on the promoter. So these genes won't be transcribed. Is that okay? Yes, we're not pissed at that repressor. He's doing a job. We don't need those genes transcribed. When would we need those genes transcribed? When we have some lactose. If we have lactose, lactose will actually bind to that repressor and pull it away. And the one I showed you simplified here to show you that. Okay? Here's regulator gene. What is the regulator gene code for? A repressor. Okay? So a repressor likes to get on the operator. So an operator, the operator is where the repressor can bind. Okay? Now, I'm okay with that because I don't need these genes. These genes over here, if transcribed and transcribed into mRNA, translated, will make enzymes to digest lactose. I don't have any lactose, so I don't care. But now, if this bacteria gets exposed to a bunch of lactose, now I want to burn these in cellular respiration. I want to digest lactose. What is lactose digested into? Lactose is a disaccharide. You know that. What is it digested into? Galactose and glucose. Good. So now I want to digest it. So now I need this gene transcribed and translated. I need the enzymes to digest lactose. Well, look what lactose does. It's called an inducer because this is an inducible operon. Because once lactose is here, it binds to the repressor and it pulls the repressor away because the repressor can only bind to one thing. So now it's binding to lactose. So now RNA polymerase can bind to the promoter, chugga, 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 make this mRNA that you see right here. This mRNA gets translated. When it gets translated, it translates it into enzymes that start digesting lactose. And when all the lactose is gone, we don't need it anymore, the repressor gets back on there. That's called an inducible operon. That operon is in the off position because the repressor is in the on position. You absorbing that? Okay, that's an inducible. We have a way of turning it on, but we only turn it on when you need it. Right now, the lights in your bedroom should be off because you don't need them. You will be the inducer. When you go into your bedroom, you will flip on the lights because now you need them. Does that make sense? Okay, so and going back to that other picture I showed you, okay, so here's an active repressor. It's on the operator. So then in the second picture here, lactose bound to the repressor, removed it. Now RNA polymerase gets on the promoter, transcribed into mRNA. This will be translated into enzymes that can help digest lactose. Now, another example, okay, this was an inducible. What's the opposite of inducible? Repressible. So this is one where your repressor is in the off position. It's built off, okay? And if it's built off, then your operon is on. And so RNA polymerase is able to get on the promoter. It's transcribing these genes. And when it's translated into a protein, it builds tryptophan. Once I have enough tryptophan and I don't need any more tryptophan, I don't want to keep making it because it's wasting. So now the tryptophan will bind to our very inactive repressor and make our repressor active, turn our repressor on, so our operon is off. Because I don't need any more tryptophan. And when I run out of tryptophan, I run out of co-repressor. It acts as a co-repressor. And so therefore I can, that's okay, I ran out of tryptophan, I want to turn that operon back on so I can make more of it. Again, this is dealing with a repressor. Anytime you're dealing with a repressor, what kind of control is this? Negative control. Now, we had an analogy, though, because we knew that glucose is the preferred substance. So one way to inhibit the formation of turning on the lac operon, even if lactose is present, is we only turn on the lac operon if we have lactose, right, for starters, and if we don't have any glucose. How will we know if we don't have any glucose? Well, when we run out of glucose, we start building a bunch of, we have cyclic AMP, monophosphate, right? So when CAMP is high, glucose is down because they ate it all. Okay, when CAMP is high, glucose is down. Now I have a signal to tell me, hey, we don't have any glucose, so we better start, digesting some lactose here. But we can only digest that lactose if there's lactose present. Because by having lactose there, right, we'll remove the repressor. And if by not having any glucose, cyclic AMP can help because in this case, cyclic AMP will bind to an inactive CAP. CAP is an example of a transcription factor. The cyclic AMP will bind to this transcription factor and activate it, and now it acts like the Boy Scout and helps RNA polymerase bind to the promoter. So with RNA polymerase getting help and the repressor being gone because lactose is gone, now we're really going to town and we're transcribing the genes to digest that lactose. If we had no repressor because lactose is there, right? So it could get on the promoter. It just doesn't get on the promoter very well unless we activate our cap. So that inhibits it. Yes?
Is cap considered a transcription
yes.
control or a transcription
factor?
Okay.
Yeah.
What's the difference between activators and factors?
Activators help factors. Activators are like cyclic AMP.
And then the factor would be
the actual thing that helps RNA polymerase bind. And that factor can be a conglomerate of several different things. This is like super simplified. Okay, so let's level it up into, when you talk about that in eukaryotic cells, it's something like this, okay? So here we have an activator bound to an enhancer region, and then the transcription factors, and see how it's multiple things right here, are all bound together, and then that helps RNA polymerase bind. So it's not just one thing. It's like you have to have this, and this all there, and then we'll transcribe that gene. And think about it. That's a highly evolved, adaptive measure, right? Because we have so many genes. If you transcribe the wrong genes at the wrong time, we're in a lot of trouble. So that's why we want to have all of these things in place before we start transcribing and translating. Okay, because these are all different levels of control. And that's what we talked about when we said different levels of control. We said, okay, remember this? This showed you all the different levels where you can have control. And so we said, is the DNA wound on that spool or not? Now we're not even concerned what genes do you have. I can't even get access to your genes. I don't know if it's a tall pea plant or a short pea plant allele because I don't even have access to it, right? Because it's all tightly wound on that histone. And things that unwind and allow that DNA to be there, that's where it's called epigenetic inheritance, because epi means what? Above, okay? So this is above. It's like it doesn't even matter. That gene can be completely masked and put away. I just can't even have access to it because it's all wound on those histone proteins. And we talked about how on the tails of those histones, there are places where things can bind. And you can also bind right onto the DNA. DNA methylation, right? We talked about DNA methylation, and it can coil up even more. Or you can have a histone tail and have something on it and might relax it. These are just underneath the umbrellas of controlling whether the DNA is available. Is it heterochromatin? Is it euchromatin? And we looked at heterochromatin being darkly stained. We looked at bar bodies being an example of an X chromosome being heterochromatin and how we are mosaics. Then we looked at just transcription in general and transcription factors, which are more likely, and it's really more like this and really more like this. And that's the biggest point of control for most control is, can RNA polymerase get on that promoter and do its job? Once we're done with that, we can cap it, we can tail it, we can modify it. It has to get out of the nucleus. Whether or not it gets bound to a ribosome or not, these are all things that can interfere with that. Yes?
In post-transcriptional control, what's the difference between microRNAs and
small interfering? Any RNA, sometimes the small interfering just do one local gene, and some control several different ones, just how big of an impact they have. I'm not going to have you differentiate between that.
Okay.
Yeah. RNAs should just get in the way, because RNA are what? Single or double? And your mRNA is single-stranded, right? So then they can hook up and become double-stranded. If they're double-stranded, then there's no place for tRNAs to land, right? Or it may not be able to hook up with the ribosome. Okay, and then we talked about post-transcriptional control. We talked about introns, and we talked about exons. And then remember we had a jumping gene that jumped out on the wrong one. Okay, so transposomes are jumping genes. So that transposomes are when it happens at the DNA level, right? Snurping happens at the RNA level, right? The transposomes could happen in the DNA level. Snurping can happen in the RNA level. And we can reorganize genes or remove some. And then we talked about folding. Post-translational control is how translational control is when it tries to get on the ribosome itself. Okay, can it do that? Can it make its protein? And then post-translational control is how is it folded afterwards? Let me look. Black operon, translational control, positive, negative control, TF active. So we hit all these stressors, right? Okay, so I'm just going to keep reviewing, but we hit the ones that were on your list that you want to make sure I went over. Okay, then we said, okay, well, how can we have mess-ups? Well, if the DNA is messed up, there's a chance then that the RNA is going to be messed up, right? Unless it's a what mutation? Silent mutation. If you end up getting the same amino acid, it's okay. Because some of those amino acids can be coded for in several different ways. So we're cool with it. I mean, it's a mutation. Yeah, it's not the DNA that we intended, but we get the same amino acid, then we're fine. Where we get really concerned, what are some ones that are really could cause a problem? Frame shift causes a ton of problems, right? Because then all of our codons are off. Because if we eliminate one, we looked at that with the like 3-letter words here. My mouse is not, go away, I tell you. Okay, I'll just do it this way. All right, so in this case, we're changed these to model it better for your understanding into 3-letter words. And if you take out one letter, then you have a frame shift. What we're reading then is this first codon is now THB instead of THE, and it throws every single codon off. That's a really bad way to mess up your DNA. What's another bad way to mess up your DNA? I heard you say it.
Making a stop.
Yeah. If when you alter your third nucleotide or a first one, and all of a sudden now you have a stop codon, you have an incomplete protein. It's not going to have all of the amino acids. It's been interrupted right in the middle of your sentence, right? So you're not going to complete that thought, that protein thought. Okay. What's another thing that can go wrong? Yes.
I actually have a question about one of the mutations of protein activity.
Yes. Let's talk about it.
For the non-functional proteins, what exactly is that?
Okay, so a non-functional protein, what you're going to have in a situation there is if you have the wrong amino acid in there, then it could fold wrong, and then if it's folded wrong, it could be non-functional.
Okay.
Yeah, and the example we talked about is we could say, like, when you make hemoglobin incorrectly and it folds wrong, then it doesn't have the red blood cell, which is made out of 250 million molecules of misfolded hemoglobin. It's not going to have its biconcave disc shape. It's going to have that sickle shape. But we looked at chlorine channels, right? And remember what happens when you have a bad chlorine channel? Remember what the cystic fibrosis. So you have one misfolded protein for your chlorine, all your chlorine channels. It's a major problem, right? A cystic fibrosis, and it's not just lungs, it's kidney and liver and all those other places too. It causes all kinds of problems. Okay, so let's keep going on that. We talked about like in a series, like phenylalanine, that can build up, you can get PKU, but also if you don't have that enzyme, you're not going to have tyrosine. And let's say you do have this enzyme A works, now we have tyrosine, but if we're missing this one, enzyme B, you're going to convert tyrosine to melanin, which is our pigment in our skin. So if we don't have that enzyme, then we have what? Yeah. So you would be an albino. So you have this one thing here that you're coding for to make phenylalanine, or you could get it from your food, obviously. But if you mess up this enzyme, you have this huge problem. If you mess up this enzyme, you have this huge problem. And then we said, what if we had problems, remember, with what were the two big genes we were concerned about with cancer? Tumor suppressor genes. Who's a tumor suppressor gene? P53. He says, no, let's fix this. And if we can't fix this, we kill you. Through what process? Apoptosis. What's another cancer gene we're worried about? Proto-oncogenes mutating into what? Oncogenes. And when they do that, they're saying yes to the cell cycle. What's our example there? RAS, like stepping on the gas, right? And so we said that can cause problems as well. And then we talked about that. We're good. And that was the end. Yay! Do you feel smart? Yes. Now, we've talked about this, so go and look up your essays and try to write your essays on your own, and then you can come back to me in another review and we can discuss them. All right? Aren't you glad you don't have a test with me this week like you do in all your other classes? Or do you want to have one for fun? Okay. All right, you're very smart and I'm proud of you. And if you're watching this late at night, have a piece of toast.